article focuses on the inhibition of AXL, a gene associated with tumor cell growth, which in turn activates HER3, another cancerous RTK, receptor protein tyrosine kinase. This links directly to our cell signaling lectures, where we unpack the function of RTKs. RTKs are enzymes that phosphorylate tyrosine residues on their substrates. These enzymes regulate growth, division, and differentiation, among other key roles within the cell. All RTKs comprise of an extracellular domain containing a ligand binding site, a single hydrophobic transmembrane alpha helix, and a cytoplasmic domain, which includes a region with protein tyrosine kinase activity. With reference to our article, HER3 is activated when a ligand binds to an RTK's extracellular binding site. The receptor is then ready to combine or dimerize with other HER receptors, forming cross-linked dimers. Once linked, the receptors are activated and can phosphorylate the intracellular portion. This allows signaling molecules to bind to the receptor's docking sites. Many signaling pathways can subsequently be activated as different proteins bind to the receptor. In the HER3 pathway, this includes activating PI3K, which may prevent apoptosis of cancer cells. The HER receptors also exert their effects through the recruitment and activation of signaling molecules of the MAP kinase pathway, abbreviated MAPK. We learn about the MAPK pathway in our cell signaling lectures. Once the tyrosine residues are phosphorylated, GRB2 can bind to the phosphorylated RTK. After a conformational change, the protein SOS is then able to bind to the protein RAS. RAS plays a critical role in signal transduction of cell growth and differentiation. SOS catalyzes the exchange of the RAS-bound GDP to GTP, thereby activating it. In its active GTP-bound state, RAS is able to bind to effector proteins, like RAF. The kinase RAF is one of the most important effectors of RAS. Active RAF phosphorylates and activates MEK, which in turn phosphorylates and activates the kinase ERK. Finally, the kinase cascade leads to the activation of transcription factors, which will carry out the appropriate response. In the case of HER3, MAPK pathway signaling may lead to increased cancer cell proliferation. In a normal cell, however, the active RAS-GTP complex is inactivated shortly after its activation to shut off the MAPK pathway and avoid cancerous and other undesirable effects. RAS-GTP is deactivated by the G protein GAP. GAP binds to RAS-GTP and assists the RAS protein in the hydrolysis of GTP to GDP. GDP-bound RAS is no longer active and can no longer bind to RAF. As a result, the MAPK signaling pathway is turned off.